Jim Schnabel doing lecture 2 in a series from Robbins and Cotran Pathology textbook, ninth edition. Today we'll be looking at cellular responses. First of all, we have pathology terminology. We talk about etiology, which is a cause, and pathogenesis, which is the mechanism of disease. And when we look at disease, there are morphologic changes, which are structural alterations that characterize the disease in question, and the end results of the disease are caused by functional derangements and clinical manifestations. So the body, the cell, has a variety of different mechanisms to cope with this. We seek a homeostasis environment where we have a stable situation where entropy is retained. And if there are perturbations to that, then the organism has means of having adaptation to this. So among the adaptation forms are hypertrophy, which is an increase of size of a cell. There's also hyperplasia, which is the increase of number of the cell. So these generally go together, but there is a discrete difference between the hypertrophy and hyperplasia. Also, you have atrophy, where there's a decrease in size and activity. Size of the cell, which usually correlates with loss of activity. We usually talk about this in terms of lack of muscle use and atrophic changes in the muscle. And then finally, you have metaplasia, which is a change of cell types. You can have squamous going to glandular, or vice versa. You can have urothelial cell alterations to squamous. There are a variety of different ways in which this is accomplished. We have cell death, which is an irreversible and reversible injury types. This is a response to its progression of insults. And cell death can either be programmed or not programmed. And again, in hypertrophy, there is no change of cell number. This can be physiologic, for example, in pregnancy or exercise, or pathologic, as you would see in hypertension, where the heart enlarges. Hyperplasia can occur in concert with hypertrophy. This can be physiologic, for example, lactation, pregnancy, liver regeneration, or pathologic, you would see, for example, in pituitary hypersecretion. Atrophy, again, is reduction in organ size due to cell number decrease or cell size. It can be physiologic, for example, loss of embryonal structures, or pathologic, caused from disuse, denervation, blood supply decrease, pressure, or malnutrition. Atrophy can be also mediated by the ubiquinone proteasome pathway. And metaplasia, again, is one tissue type transformed into another type. So, for example, intestinal metaplasia in the stomach, squamous metaplasia in the lung. These are usually protective responses to the microenvironment. And this is indeed deleterious change, in some cases pre-neoplastic, which, for example, intestinal metaplasia in the stomach is such an example of this. Cell injury causes oxygen deprivation. You have reduction in oxidative phosphorylation. This leads to ischemia. Physical agents that can do this are trauma, temperature, radiation, pressure, and electric shock. Also, this can be caused by chemicals and drugs, toxins, pollutants, occupational, industrial hazards, alcohol, recreational, and therapeutic drugs. Infectious agents can also do this, viruses, fungi, bacteria, rickettsia, and parasites. Immunologic reactions, including autoimmune diseases or responses to infectious agents. Genetic derangements, which can create enzyme or deregulation defects. And also nutritional imbalances such as starvation, vitamin deficiencies, and obesity. So when you look at the progression of morphologic changes in cell death, cell function decreases, and you have, in the duration of injury, you first have the biochemical alterations which culminate in cell death. This is followed by ultrastructural changes you can see using electron microscopy or some device like that. Finally, you have light microscopic changes you can see under the microscope. And then lastly, gross morphologic changes. So these go from micro to macro changes where you ultimately will see what would be called infarct, which is death of tissue within organ. These are irreversible. The cell function loss is reversible, and in that you get the biochemical alteration. So it's reversible up to this point when you start to get the ultrastructural changes, and this is a marker of a irreversible process. So molecular changes begin with mitochondrial damage, with ATP depletion and lysosome-mediated rupture of membranes. Reversible injury has cellular swelling and fatty change. The former is due to ion pump failure, and the fatty change is due to lipid vacuoles in liver and muscle, which is a difficult concept to fully explain molecularly, but it is something that's readily seen under pathological examination. Necrosis is death of cells of tissue, and this is usually seen with nuclear fragmentation, which is called karyorrhexis, which is fragmentation of nucleic acid, and cytoplasmic loss of RNA. So the cells become progressively more red. Coagulative necrosis is dead tissue confluence after the passage of time, and the larger areas constitute an infarct. So we talk about coagulation necrosis basically being due to ischemia. There's also liquefactive necrosis, where the dead cells form a liquid viscous mass. The common example is pus. Gangrenous necrosis is a clinical term, and this is necrosis which crosses tissue planes, often seen in the leg following diabetic insult. Caseous necrosis is friable, crumbly, sort of like broken cheese, and you see this characteristically with mycobacteria infections. Fat necrosis is destruction of fat, and this combines with fat as saponification, and this is seen oftentimes in manifestations of pancreatitis. Fibrinoid necrosis is seen usually in immune reactions, and this is an important thing to notice because this creates an entire category of diseases, and you see that and you start thinking about immune-mediated entities, and this is seen in blood vessels, oftentimes in the skin, or many times in larger vessels. We talk about the extent of injury. These are related to the nature, duration, and severity of the agent. The consequences are dependent on the type, state, and adaptability of the injured cell, and that the latter of this may have a genetic component, the ability to withstand injury. The injury depends on a spectrum of the cell component susceptibility. ATP reduction is a fundamental cause of cell death. This is affected by hypoxia, mitochondrial damage, and, if applicable, toxins. The molecular mechanisms is that first there's a failure of the sodium-potassium ATPase, then oxidative phosphorylation fails, glycogen is depleted, you get anaerobic glycolysis, the calcium pump fails, and protein synthesis stops. So this is seen schematically here, where you've got extracellular calcium which enters the cell. This is an injurious agent. The mitochondria and the smooth endoplasmic reticulum relinquish their calcium stores as increased calcium, ionized calcium in the cytosol. This activates cellular enzymes, phospholipase, which decreases phospholipids, proteases, which disrupt membrane and cytoskeletal proteins, and the nuclease, which disrupts nucleic acid, ATPase, which reduces ATP. So all these effects are caused by influx of calcium and increased permeability, losing calcium from these structures, and then you get nuclear damage, membrane damage, and a loss of ATP. Free radicals accumulate. This affects oxidative phosphorylation. There's increased inflammation, peroxides, and nitrogen-free radicals. This causes DNA breaks, there's protein oxidation of side groups, and lipid peroxides. Membrane permeability is affected. There's damage by free radicals, phospholipid breakdown, and cytoskeletal damage. Hallmarks of irreversible injury include lack of oxidative phosphorylation, membrane function damage, and the latter causes leakage of substances monitored for cell death, proteins, and organ-specific enzymes. Ischemia is caused by hypoxia. This can be exacerbated by reperfusion of blood to the damaged tissues through activation of oxidation, calcium, inflammation, and complement. The chemical damage can be affected via direct injury by chemical or toxic metabolites. Apoptosis, again, is programmed cell death. This is a physiologic type to eliminate cells no longer needed. There is, however, a pathologic type, and this is caused via atrophy, infectious etiologies, protein structures, and DNA damage. There are two types. One of these occurs through the mitochondria with dissolution of its membrane. This process is inhibited by BCL2 proteins and exacerbated by BAX and BAK proteins, and these are detected by sensory proteins including BH3. Cytochrome C is released. This binds to APAF1. This is an apoptosis activating factor 1. This forms a complex which activates caspase 9, and that initiates the apoptotic process. And also there's other apoptotic mechanisms, death receptors, which are part of the TNF family. This acts through FAS. This is detected on cytotoxic T cells, and this activates caspase, and this is a cascade reaction. There are cells removed by phagocytosis. Incomplete or dysregulated apoptosis creates persistence of mutagens or intrinsic degenerative diseases. Necroptosis combines elements of apoptosis and necrosis and does not utilize caspase. There's also pyroptosis, which is cell death mediated through release of inflammasomes responding to microbial infections. Autophagy is a survival mechanism. The cell destroys itself through lysosome fusion and digestion. It is programmed into the genome. It plays a role in cancer, neurodegenerative disorders, infections, and inflammatory diseases. Intracellular accumulations can occur. These are lipids, proteins, hyaline, glycogen, and pigments. These occur with overproduction or arrest of metabolic pathways, deposition of non-metabolized substances, or inadequate removal. The deposition of lipids occurs as steatosis, which occurs fat vacuoles under the microscope, atherogenic plaques, and foam cells. And proteins, you see these accumulations in kidney disease. These are abnormal accumulations and fibrillary tangles. Hyaline is a histologic term for eosinophilic acellular material. It varies in type and location. Glycogen accumulates in metabolic diseases. The pigment can either be exogenous, such as carbon or tattoo, or endogenous. So lipofuscin in the liver is a wear and tear pigment. This is a lipoprotein polymer. Melanin is a dihydroxyphenylalanine, and hemosiderin, which is an iron protein micelle. Dystrophic calcification is an injury response. There's deposition of calcium associated with necrotic cells. Oftentimes this is a marker. It's used commonly, for example, to diagnose suspicious foci of breast cancer. Metastatic calcification, however, the physiologic response, this is deposition of calcium in tissue secondary to hypercalcemia, elevated calcium from parathyroid hormone, bone resorption, vitamin D disorders, and renal failure. In cellular aging, you have a progressive decline in cellular function and viability caused by genetic abnormalities and accumulation of damaging agents. DNA damage occurs with regularity, and this is usually repaired. Cellular senescence is genetically programmed upper limit on replicative cycles. You see telomere shortening, which arrests the cell cycle. The telomeres protect chromosomal DNA, and their loss ultimately stops replication. Other factors include tumor suppressor genes. These are activated. The protein tertiary structure is malformed. There's nutrient sensing through attenuation of insulin growth factor and increased concentration of NAD-dependent deacetylases, which lead to increased caloric intake and lowered longevity. And that's the talk for today. Thank you very much.